build these very nice firing fields of like place fields that are in a particular place, or grids that form these nice hexagonally packed grids when you have a behaving rodent foraging these open free areas. But it turns out there's a hidden code that actually allows us to determine the position of the behaving rodent much more precise than a simple rate-based model based on these firing fields would, and that is the theta phase. So it turns out that when a rodent enters these firing fields, both whether these are place cells or grid cells, there's an interesting effect that the first spikes when the rodent enters, let's say the upper one here are the place cells, the spike tends to occur relatively late compared to the very strong theta signal in the local field potential, which is actually the largest in hippocampal entorhinal cortex complex. So these are oscillations 2 to 10 hertz.
I mean, you could explain this. You say, I'm looking at the upper right-hand picture, right? You said relatively late. So the gray thing is the theta cycles?
Right, exactly. So what you see here...
what's the red and what's the black?
Right, so the red is trying to delineate the different cycles of theta.
Okay, that's theta. And then
exactly. And then above in black you see the spike times. Right? So when does the spike in the place cell occur relative to the local field potential that we can record via EEG or an electrode inside the tissue. So this is an extracellular signal of sorts, and it's big.
The sentence you said was it. I know this, but I don't understand this picture. So
yeah,
you said it appears late.
Yes.
So when you mean by late?
Right. So if this is a theta cycle, from here to there, right? So you can see this is 0.6 seconds and 1, 2, 3, 4, like 5.5 cycles. So a theta cycle is on the order of 100 milliseconds or a little bit less, which gives you like something like 10 hertz. So that's a typical frequency for theta. And what you see is that relative to the theta that you can observe sort of throughout this tissue in the local field potential, the place cells, when you first enter a place cell, fire late in the cycle. So this is the cycle from red line to red line. And then they start firing slightly earlier. And then the third, fourth, and fifth spike tend to be earlier and earlier and earlier. And so there's a so-called theta precession, meaning the cells fire rhythmically, but the place cells are actually a little bit faster than theta. And they always have a theta phase that precesses, so it becomes earlier and earlier and earlier.
Does the animal need to be moving through this period of time?
Yes, exactly.
So is it moving towards its center there?
Yeah, that's an interesting question.
So the animal stops, does the period, the difference, the lateness stays. It's really a place coding. It's not a time coding, right?
It's not exactly. That's exactly the point that Barry and Burgess made. With O'Keefe here,
so this is odd because it's labeled as time, but it's really, I'm not sure I'm right about this.
Yes.
But it's time, but it's really movement in a direction towards the classic center of the place field, right?
They actually tested this, whether it has to do anything with time, by trying to correlate the firing with all kinds of different variables, like distance from the center, time traveled throughout the whole. I can show you if you.
No, it's just verbs. This language is very simple. It's just like, what is it? What is the precession coding for? Is it precession for how far away you are from the center of this field, or is it a time-based thing?
It turns out that it's really coding for the distance relative to the center of the place field.
Got it. So it's encoding how far away from this place.
Exactly.
Okay.
They tested this rigorously. You can try to control for all kinds of things. It turns out that the theta phase is much better correlated to the location than things like the speed or the time spent in the firing field. And that holds true for grid cells and place cells. So that's why there's two graphs here, right? The above one is a place cell. The below one here is a grid cell. And you see the same theta precession in grid cells.
Does that mean if it walks straight through the field, you'd expect the theta phase to go down and then up? It would go.
Does it reverse on the other side of the field? Is that what you're saying?
Yeah, as it walks through the
Yes.
Does it
So
start late, go early, then go late again?
I think that may be what we're seeing up top
Right,
there on the
right. That's what you see here. So there's a linear. Voila. There's a, I'm sorry for like trying to fit this. Maybe I should have extracted these. So what you see here is this variable that they have PDCD, which is like a distance from the center, and they show that the spikes tend to fall on these falling lines, right? So the theta phase of the spikes are precessing. The phases relative to theta is always becoming less and less and less and less throughout walking through the entire field from start to the exit of the field.
That's not what Marcus said, right?
So it's not going back after it has passed the midpoint, but instead it's continuous precessing. So the earliest spiking you will find is when a rodent is just about to exit the firing field.
So that implies there is a directionality to the signal. It's not like you can't just encode how distant the animal is. That was the question I had earlier. This says that there's a distinction between which way there's a directional preference to this.
That is a good question. It turns out...
But isn't that what that suggests?
No, because if that was true, you would expect the precession to go the other way when you enter the place field from a different angle, like the opposite direction. That is actually not true.
No, but
the
but
phase
my
always
point is,
precesses.
if here's my place field and I'm approaching from here, and I have this, it's early, and then now it's not early, and then
It's late and it's becoming early.
well, late
Right.
becomes whatever you want to
Right.
call it. I'm sorry, late, and now it's right on the peak, right? And then the question is, if I continue on, you're saying the precession keeps moving in the same direction. Where what Marcus was asking, would it just, if I was symmetrical, it would just start getting late again?
Right.
And so the fact that you're saying no, it doesn't get late again. It continues its drift in the same direction.
Yes.
Says that cell is encoding more than just a position from here. It's encoding a position and a direction.
A direction
It's not
relative.
just a distance, it's
Yeah.
more
Right.
than that.
Right,
Yeah.
but not an absolute direction. A direction relative to the center of the firing field. So
my
you
point
can
is-
you can come from any angle to the firing field. It's always going to precess. So you can't tell the direction by the direction of the precessing.
But what if I
Precessing is
didn't?
always-
What if I didn't actually move? What if the animal wakes up, looks around, and I have a place.
Theta disappears when you don't move.
Okay, it disappears completely.
No, but it's highly related to movement. And in fact, there's even a correlation between an animal's running speed
I thought the
and
frequency
theta.
of theta changed when you move.
Yes.
But
Yes, but also the amplitude of it. So the importance of theta is really to moving. Doing consummatory behavior, I don't think you see much in terms of.
So this is a really important distinction, I think.
Mm-hmm.
This is a huge clues, I guess.
Mhm.
You're coming in, it doesn't matter what angle you're coming in from, but if you're moving towards the place field, you're going to see the same precession in all cases. If you move past it, you're going to see the same thing.
Mm.
But it doesn't matter which direction you're coming from. And you're saying this is my question, I said if you stopped, you said it would maintain its phase shift, but if I just woke up there, then maybe it's meaningless because there's no theta. Is that right?
I would agree with that. Yes.
Okay, well, I'm trying to play what you said.
Right. No, I'm learning about this myself.
Okay,
right?
okay. Well, that's all right to say we're not certain, but it's an interesting distinction. It's kind of an odd encoding.
So
I have a quick question
Right.
about the center. They talk about the center of the firing fields. Did they say how they determine what the center of the firing field is?
yeah, they have some. I mean, they have like I don't know, like if you look at this map here, right? They have like 100 spikes or something per firing field here. I don't know, maybe it's just 30, I can't tell by the dots, but it's enough to build a heat map like this and establish a mathematical model that determines the peak, the center, right? So then they use that for their math.
So again, if I wanted to lay over a phase precession thing here, or shift around those red blobs, I would see at the center, if I could have a little heat map about how much it's precessed or whatever, I would see the color at the center, and then it would sort of fade off, and
Right.
it would also encode the direction.
Right.
So those dots in theory would occur at different phases of the theta cycle.
Yes.
Okay.
So the reason why I mention that is just to point out that this is a reliable finding. true for grid cells and place cells, that the theta is massive in hippocampus and entorhinal cortex, so we somehow need to explain what's going on there, why is this so important for it. And like the amplitude is also larger than anywhere else in the brain for theta, so it seems to be like a really important mechanism. And so that's why if you want to build a grid cell place cell models, maybe you want to build a model that actually accounts for these things, rather than just a model that generates a nice grid.
Well,
right?
let me ask a stupid question. Why do we need to do it? Why can't we? Why is it important to understand how these things emerge? Why can't we just use what we've learned to build systems?
Yes, well, in part is because we have a desire to understand the brain and not just build a system that has a grid. I mean, like, there's
it's pure research. So
yeah, it's lots of ways to build a
you
grid.
just say, we want to understand how the brain does it.
But do we think this is necessary for intelligence, the emergence of this grid? Is that part
We don't
of
know.
intelligence?
We don't know, and I've learned to never say something's not important. I mean,
Right.
you just,
That's fair enough.
after you understand it, you could say it's not important.
Yeah,
but
okay.
you can say I can't figure it out now, so I won't think about it now. But this seems to be very key, because as I've pointed out multiple times, it's possible this theta is involved in movement, in controlling behaviors, and indicating direction of behaviors, and all. So this could be key to building machines that move.
Yeah.
And I thought you mentioned something pretty important early on, which is, determining location precisely is important. And this is a mechanism through which you can detect your location much more precisely than using firing fields.
Because you get an exact distance to the center of the firing field.
To me,
So this reduces maybe the need for in grid cell, the number of modules you might have and stuff like that.
Exactly. So it's additional information, and so that reduces the need for so many.
To me, the big things I just mentioned, the really big things is like this huge gap in our understanding between how behavior is represented and generated in the brain. And we talk about, we have some behavioral signal, motor signal. Well, it may be that signal is this theta phase, and so to me, this is I would bet it's much more likely than not that this is a critical component about how you build systems that actually. Not that we have to have a theta cycle, but we need to understand exactly what's going on at least.
Yeah.
Right. So most of the grid cells we find in medial and entorhinal cortex, particularly the layer 2 stellate cells. The interesting thing about these...
Say that sentence again, much of what we find?
Most of the highest grid cell density, the most grid cells,
okay,
right? And actually where they were found first was in medial entorhinal cortex. We've since found a couple in other brain areas in much lower density, like the paracerviculum and like neighboring brain areas too. But that's where we have the highest density of these grid cells. So an effort is to try to really understand what these layer 2 stellate cells in medial entorhinal cortex are doing. And it turns out these stellate cells really have some interesting properties. So for one, they have...
These are grid cells, right?
These are grid cells, or like all the grid cells typically are layer 2 stellate cells. There are some exceptions. And so what we're really trying to understand is these cells, and they're interesting because they have what is called an intrinsic membrane potential oscillation, which is actually independent of the global theta that sort of syncs up hippocampus and entorhinal cortex with this global rhythmicity. So when you record the membrane potential of a layer 2 stellate cell, there's a sub-threshold oscillation that is quite prominent, and it turns out to, I think it's like 8.5 hertz or something. It actually changes a little bit along the dorsoventral gradient, which is actually going to be important for the model. It's worked by a colleague of mine from Stockholm, actually, Eric Francine, who worked with Mike Hasselmore, I think, on this. Anyways, we're getting lost in like tiny things here, but it's just interesting to note that somehow these frequencies are reflected, obviously, in some of these cell properties. So what I want to start with is the first oscillatory inference model for trying to explain a grid cell, or rather a place cell, on a linear track.
Could I ask a question? Are those subthreshold oscillations, are they all correlated, or each one is independent, each cell has an independent one?
That's a good question, to which extent they are synchronized or not. As far as I understand, they come in groups. So you might have cells that have synced up in various ways. But that goes to the continuous attractor models. It's like a bit.
Okay.
That'll be the walking in assumption that they're synchronized.
All right, so first figure of the preceding paper. Rather simple idea to try to explain this processing of the face of a spike is to build a model that says, look, if I have a cell, like a layer 2 stellate cell in medial entorhinal cortex, and this cell has some intrinsic oscillation with some frequency omega s, right? So somatic fluctuation, right? That is somehow sinusoidal and is described here with a cosine and some amplitude as. Then one way to explain this firing is to say, look, if I add to this cell, which already has an intrinsic membrane potential oscillation, an input that on the dendrite, which is also oscillatory, so with some frequency omega d and some slight phase offset, then I can build a model that explains much of this. So under the assumption that that dendritic oscillation is modulated by the speed of the behaving animal. So if the animal goes fast, I should probably point out this is the speed, then the dendritic frequency will be noticeably faster than the somatic frequency. So this is a linear track, so there's only one direction, and so the speed is in this case, right, it's 1d is the speed in one direction. Then it turns out this theta precession automatically shows up if you just say all that happens here is a thresholded sum of these two inputs. You have the intrinsic oscillation, and on top of that is a dendritic input that is a slightly higher speed.
When you're moving.
When you're moving.
When you're not moving, it's the same.
Picture of those.
Go ahead.
Right. So
Is that right?
so if you,
I think that's.
I'm not sure exactly
Okay.
what happens when you're not moving, so I
Well,
can't
okay,
speculate.
I'll keep going. I think
Right.
that's what-
So this model presumes that there's always some speed at which you're moving. So the dendritic frequency is always, the same as the somatic or slightly higher. And so if you are moving and you threshold that thing at 0, then you will get spikes. And these spikes will be theta rhythmic, but they will be slightly faster than theta, which means that they go to earlier and earlier relative to the phases of theta. How does this come about? It's actually relatively straightforward math that you can do here. So if you just sum these two different inputs, very simplest possible model, you're just summing the dendritic input with the somatic oscillation. What you get is that that there are really two components to the oscillation that you get out. One is what we call a carrier, and that gets multiplied with a different cosine, which we might call the envelope. And it turns out so this is just doing the math of summing these up, right? And doing some manipulation math-wise. And so under a couple of simplifying assumptions, you can see that, for example, if the amplitude of these two oscillations was the same, then this term here would cancel out. This one here, omega s plus omega d over 2, that is actually an average omega. So I'm going to denote it like this. And this omega d minus omega s over 2, that's sort of like a delta omega half. So this is the average omega between the somatic and the dendritic, and this is half of the difference. And this shows up, so if you do this, you see this here, the sum in black of these two oscillations. The dendritic oscillation is a little bit faster than the somatic. And what you get then is this wave here, which has an envelope that is very slow because it's the difference between the two oscillations. And you have a carrier that is a little bit faster than theta by this equation here. And if you just threshold that at 0, that will give you all the spike times here, and then they will nicely precess.
Doesn't this say that the faster you move, the faster the precession is going to be?
But the faster you'll go through those big.
If
Yeah.
you move faster, then the dendrite will speed up, and then yes, you're gonna get different precession.
Is it
Yes.
speed related or is it distance related then?
So my understanding of this theory, I'm sorry.
Or is it, it's really hard to disentangle the two?
Yeah, it's a little bit hard. I don't think I can give you
The way
straight about
I
that.
understood this, and Marcus, you probably know better than I, is that you got these two frequencies. If they're the same, then nothing changes. You're stationary here. The cell's either firing or not firing, or it's either at some position. But when you start moving forward, you get one of those frequencies speeds up a little bit, and now you'll start getting this sort of in your music. You hear this wah 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 sound, and the two notes are not exactly the same. And if you speed up more, you go wah 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 wah. So it's basically those wah wah wahs are your grid cell places, and
Okay.
so. If you stop moving, then the frequency should go back to the same frequency. That's what I was thinking. And then you stay at that one point. I mean, you can understand it all without any of the math. Just imagine these things lining up just like this.
Right.
So I think it encodes, my understanding of this is that the change in frequency represents the speed at which you're moving. And by physics, that sort of applies how far you've gone. So you're doing path integration by speed.
Yeah,
And
exactly.
those big waves there are basically your centers of your grid cell firing fields.
Right. And essentially the integration happens through the phase shift, right?
Yeah.
Right.
So two important caveats. So while this is a very nice model that just turns out to mathematically produce some things that are sort of been observed, there's already two caveats that I want to point out. I don't want to solve them yet, but I want to make you aware of it. One problem of this model is that if it is indeed true that the dendritic oscillation is the somatic plus some constant times the speed, that also means that if you were running the other direction, so if the speed were to turn negative and the dendritic oscillation would actually be slower than the somatic oscillation, then you would see not phase precession, but reverse phase precession, which has not been observed experimentally. So there's a problem with this model. There's a way to fix that, but we have to see whether we can get to
That
that.
seems like a fundamental issue.
Yes, it is
Yeah.
a fundamental issue, and it's best fixed mathematically by assuming multiplications instead of additions, which has to do with supralinear summation, but I will get to that better in a slightly bigger model.
But these are dendritic oscillations. That
Mhm.
means they're caused by some input coming in
Yes.
to the dendrite.
So it is well
How are you
known.
going to have negative input? Positive suddenly turn to negative.
So it is well known that these intrinsic membrane potential oscillations, so what these neurons do without any input, right, is that if you they have this intrinsic oscillation that is sub-threshold, so it doesn't make the neuron spike rhythmically. But obviously, if you add input to that, you can reveal it, and it turns out that most of these intrinsic membrane potential oscillations are sensitive to excitatory input. Exactly describing this, that if you provide excitatory input, these oscillators will speed up. So in fact, you don't necessarily need a circuit that generates some kind of oscillation if you have cells that do that by themselves, and if thresholded at some value will produce rhythmic spikes.
This is a very high level conceptual thinking about this.
Mhm.
The problem is it says, this thing can only tell you if you're going in one direction, right? It can't say you're going backwards, right? But we have to include the whole concept of orientation in the navigation of the animal, head direction cells, orientation cells. And so in theory, that whole problem could be shifted to another part of the neural machinery here that involves orientation. That is, these particular mechanism, it just has to say, well, I don't know which way I'm going, but it's like I'm always going forward, and someone else is going to figure out what the orientation of that is. I can't walk backwards in some sense. I can only walk forward.
Like it's an absolute
But if
value.
I want to go backwards, I sort of have to turn my orientation and go forward.
Right.
So then it becomes not a substantial. Then it says, oh yeah, we have to solve the orientation problem at the same time. I mean, it's not a completely fundamental problem with the theory. It's just that hey, you've got to solve this orientation issue.
So that is one issue, and if we have time, I will also show the solution to that. And the other issue that I want you to be aware of, that there is a slight problem when we apply this to 2D in particular. One is the phase noise, so that these phases of these theta oscillations, they need to like link up in order to make any sense. What do I mean by that? So if you want to now build a 2D grid with oscillators in different directions, so maybe you want 3 and at exactly 60 degrees, how and why, we'll talk about that, and if it actually needs to be that strict. Then it's relatively easy to draw the first two. We can really put them anywhere. The problem, when you now want to make this into a regular grid and you have a third component, it actually needs to be such that the phase of the third component actually falls exactly on the overlapping of the preceding two. So how do you sync up the phase of this third component such that it doesn't intersect somewhere in between? Like this would be kind of useless for a grid, right? So you want all of these components to nicely sum up. So there's one problem of like resetting the theta phase, making sure that these membrane potential oscillators that you attach at the dendrites, now it's not 1, but instead it's 3, that they're actually in phase.
Would this be another vote for really having one-dimensional grids or modules?
Yes.
Okay.
Because fundamentally, the argument that this idea would pose, right? So this construction essentially says there are one-dimensional oscillators who only care about movement in one direction, and they oscillate at theta plus whatever component in that direction that unit detects, right? By using some speed input and some orientation input. You can argue about how these work. But the point of this mechanism is that you get, now if you build such a model and you say there's 3 of those and you have a behaving rodent, so like you see like the simulated mouse here is heading to the right down. So it's kind of heading in the same direction, heading orthogonal to the blue oscillator. So that's why the blue oscillator is at a low speed, where it is heading in the direction of the green and the red one. So the red and the green oscillators have a slightly higher frequency. And what you will see in terms of so this is the somatic theta. And if you sum all of phase precession grids in 2D and actually make that exact firing timing an indication of the distance from the center of your grid
And this
field.
doesn't solve the reversing direction problem because...
No, not yet.
Not yet, yeah.
Not yet.
This doesn't, you can't tell me which way I'm going.
Right, and it also doesn't explain how do I make sure that, for example, if I implemented the red and the green components first, how to make sure that the blue one actually lines up and intersections at the intersection points of the previous two, right? So the phase synchronization problem. But it is a very nice model that will yield a grid, and it will explain many of these things with the theta, about why there's big LFP in there, why it will explain precession, and if you tune it right, obviously you can get the right firing rates and whatnot.
Could I try to tell you 2 things that you mentioned together?
Mm-hmm.
The stellate cells you were talking about, oscillations, and this. There's another way of looking at that. Rather than summing, if they multiply, it becomes a class. If it's sine waves, it's a classical E thing where you produce sum and differences of the frequencies as well.
Right.
So I'm wondering, in that formulation, do you have a better handling of the phase problem?
Yes, you do, and that's actually part of the solution. I'm trying to skip the math on what it would look like to actually multiply up 3 different oscillations at 3 different angles together with a somatic theta, like to save you that. It's complicated enough for like a 1D case.
Right.
But it's true that you can solve the problem of directionality of the precession, so making it always precess rather than go the other way, by making it multiplicative rather than additive, which requires a lot of requires very superlinear adding, which means that you need to do this at cells that have very thick dendrites of sorts. Turns out that the medial entorhinal cortex layer 2 stellate cells have exactly that. So people have been looking at the morphology of like these cells. Do I have a picture here? This one or...
One of the side effects of having that sums and differences is that it produces a Vernier-like effect,
Why
so
does it not?
you increase your precision.
Why does this not change?
So that's what I thought was kind of interesting when you showed me those first two diagrams.
Right. Here we go. No, it's actually not what I meant. No matter. No matter. The point is, I can show this paper. It's fine. So people have been looking at that, and so this is just an abstract from a different hippocampus paper, actually a lot older, right? Where they were looking at the morphological characteristics of these, putative grid cells in layer 2. So rat, medial, entorhinal cortex, and they have in this abstract here, they say, right, all neurons, so 65% of the total cells recorded, that develop sustained subthreshold sinusoidal membrane potential oscillations, so that's exactly this mechanism, this theta mechanism, were found to have stellate appearance, so it's sort of like a property of these stellate things. And neurons that did not exhibit oscillatory behavior had either pyramidal-like or horizontal cell morphology. Stellate cells had multiple thick primary dendrites. I think they said it was like something on the order of like 4 to 8. Now, if you want to build a very nice model with 4 to 8 inputs, you can ask, okay, how does this model change? So this is like 3, and so only going in 3 directions. But if you want to build a proper model, then you might want more. And so you can actually iteratively build a model. So maybe we just start with...
Why would you want more?
This. Well, I will show in a second. So the easiest case, right, if you just have an oscillator in one direction, right, and you now want to see like where it fires, you get something like this. So in the direction of so that the direction that this cell is sensitive to is to the right. So if you go orthogonal to it, right, then the dendritic oscillation will be the same as the somatic oscillation according to this equation. And so, in fact, so what you get then is that the average oscillation, right, the carrier frequency will just be a regular theta, so will not be faster, right? And that this envelope goes away. So, in fact, because they are the same, so this goes to 0, right? And so what you get then is just a constant oscillation where, in the direction orthogonal to this one component, right? Only one dendritic oscillator. There's no change. Whereas in the other direction, well, there's actually a couple more. So if you go to the...
Can I ask, this is important. I'm sorry to interrupt.
No worries.
So we have this theta frequency, and then we have these oscillations in the dendrites.
Mm-hmm.
Is this suggesting that the oscillations in the dendrites are independently representing different? I mean, they are the motion. They are the speed. Those are the ones that change the speed, or is it the theta one that changes the speed?
So the theta of the dendritic input changes with speed in specific directions.
You got these multiple dendrites.
Yes.
You got multiple dendrites in the stellate cells.
Mm-hmm.
They are representing different things.
Yeah, they have oscillations that code for, with their oscillation frequency, the movement speed in different directions.
All right, there's a lot of words. So if I start moving, so let's say I have 3 dendrites.
Yes.
And I start moving, and they're representing orthogonal direction or something like that, right? If I start moving in one thing, then the oscillation in that dendrite would change in one but not in the other 2. And then if I went in a different orthogonal direction, the oscillation in that new one would change.
Exactly.
Then is the theta changing at all?
No.
Does the background constant?
Stays the same.
So
stays theta.
So basically what now we're saying is that the encoding of direction, if you will, is on a dendrite by dendrite branch basis on a particular cell. it's interesting then why I always thought the theta sped up when you moved. But here you're saying that the dendritic oscillation would speed up when I move. Well, which is it? Both?
There's two thetas here, right? One is the somatic theta, and one is the dendritic one. And there's
when you say somatic theta, I'm thinking about the one that goes. I'm always thinking about between the thalamus and the cortex. Well, I'm thinking thalamus and cortex. We're not doing that here.
In the cell body.
In the cell body, there's one.
There's one.
That's the somatic.
Okay, so you got this
the
one.
dendritic one is what speeds up with or changes with speed.
And when does the somatic one ever change? Does it ever change?
Yeah, for example, it changes along the dorsoventral gradient. There's a so-called H current.
No, but on an individual cell.
In this model, does it change at all?
No.
Okay.
I mean, this explains one module, right?
Yeah.
So it has a stable scale, right? And so that is set by the somatic theta.
So the somatic theta is the carrier? Does that make sense?
In the sense that the dendritic one is the same plus some value, yes. Right? The carrier will be the average frequency between the dendritic and the somatic.
So now the question is well, how would a dendrite know, how would it develop this orientation preference?
I will get to exactly
that's
that.
what I was asking
There's
earlier.
a
It must be getting inputs from somewhere.
Right. There's a self-organizing
And those inputs
mechanism that can do that.
I mean, I was thinking
Get to
about the proposal I talked before the break in the cortex, and again, this is not cortex, that this idea of having inputs that are directionally sensitive, sensory inputs that are directionally sensitive, is a way of defining these orientations and how you could. But I was thinking that it was on a cell by cell basis. This is on a dendrite by dendrite basis, which is a little weird. So okay,
It's more compact.
But it's not clear how that would come about.
Right. I will talk about that.
Okay.
So let's step through this, right? So what they're building here, I want to talk about the column A, right? So there's one dendritic oscillator. And all that is shown here is sort of a simulated animal running from the lower left corner in every possible angle out to explore this field, right? That's why you see like these nice, curved lines there. And it turns out that when you go towards the right, then the dendritic oscillator will be faster. So there will be a carrier wave that is actually faster. So it turns out these circles there, they're not actually circles. It turns out that there's about 30 waves to the right and about 25.
I don't know which circle
So if you count the numbers of
ellipses.
which box are you talking about?
right.
Just point to the box.
Right. So this box here, right?
We got the first one. We're done with that one. Now we're talking about the circles.
Yes.
Which
So
one?
so
you don't zoom out again.
okay.
Right?
No, these circles here, right? So it looks like quarter circles.
Oh, that?
Right,
Oh.
but it turns out it's not, right? Because of course when you're running in the preferred direction this way, then the carrier will be faster. So in this direction it's I think like 25 circles or something. In this direction it's 30. But what you will get for the fact that you now have this superposition of the regular theta and a faster component, is that you will get an envelope. This thing will no longer be zero. And so that means that your firing rate is now going up and down and up and down and up and down. And your theta phase is proceeding and proceeding and proceeding whenever you are in a firing field. Right? So what you get here are not grid cells. This is clearly not a grid, but it's a band cell.
Yeah.
Right? So if you instead of sort of an idealized mathematical agent that runs from the lower left to any point on this map, you have a random agent forage on this map, you get something like this, and then when you run a boxcar over that to filter the spikes, you get these very nice bands.
This is observed in experimental.
Actually, it's been observed, right? It's one of the things on our list here somewhere. Where is it?
Band
yeah, here it
cells,
is, right?
fifth
Band
band,
cells, right? Should like start ticking off all of these things, right? So, so that is interesting. But in order to get a grid, you actually need to add more dendritic oscillators. So it turns out you don't need something like 6 dendritic oscillators at perfect 60 degrees to get a grid. It turns out.
Do a favor for me, Florian. I find every time you zoom in and out, it makes it harder. I can see the whole thing without zooming out. All you can just need to do is just point to the or say column
right.
C or something
okay.
like that.
Okay. So
That's
this is big enough.
Say column C,
Right.
whatever.
So now we are adding an additional oscillator at 60 degrees. So
This is B.
For which,
This
does
is
it
B.
be?
this is B1, right?
Yeah.
And we lap the agent 1. And you already see a grid.
Yeah.
If you add a third component at 120 degrees in C, right, you will see a sharper grid. These circular, they actually have like a slight elliptical tinge to these firing fields. They get finally very nicely circular once you arrive at the column D, which
They
is...
look pretty circular in B3, but I don't know why they are. It seems like they shouldn't be.
B3?
Yeah, with two oscillators, they look really circular, but when you go to three oscillators, they look elliptical, and then you go four oscillators,
Right.
I mean...
I think that has to do with the fact that you go from the lower left in this case.
It doesn't matter.
Right. The details also don't really matter. The point is when you add components at slightly different angles, right, you get a nice grid.
You have more precision.
Right, and you get more precision. The grid gets sharper, right? And so you see here in the different columns, 1 oscillator, 2 oscillators, 3 oscillators, and D is 6 oscillators, so perfectly 60 degrees separated. So this is a very ideal model of sorts.
You know, this is all being said done in one neuron.
Mm-hmm.
Why couldn't this be done in a bunch of separate neurons, and then someone sampling from all of them, and then that's what I'm seeing? You know, what if I had a whole bunch of why couldn't I just have a whole bunch of 1D band cells, and then I have another cell which samples from all of them, and then wouldn't it look like D? Am I missing something there? Is it,
Yeah, essentially your grid cell is doing exactly that. It is sampling, it's putting the different 1D inputs
yeah, but
together.
what I find a little bit hard to, not hard to accept, but less likely is that the different dendrites of a particular stellate cell are actually running at these very precise different orientations. That's hard for me to imagine.
That's the number one critique of the model that's coming.
Yeah, so it's much easier to imagine a bunch of band cells, and they're all just doing their thing, and then I have another cell which is samples from them.
But then you wouldn't be able to explain the theta precession stuff.
I wouldn't.
Because any given cell would need to get, sooner and sooner.
But all the band cells have precession.
Not in 2D, I don't know.
Well, they do because they have this dual oscillator
You
models,
can
right?
see it. I don't know the colors encoding that, or not, but they would. The single oscillator model has precession. So the A would have precession. I can't be certain that if I sampled a whole bunch of those that I think I would also get the precession.
But grid cells, do you get precession for all directions?
Yes.
So
It
you
doesn't
wouldn't
matter
get- you
which
wouldn't get that
direction
in the
you
band cell.
enter the firing field, the theta phase of the spikes that you can read out will process.
Band cells, you would only get it in one direction, you wouldn't get it in
Yeah,
all different
fine.
directions.
Fine. Is that a problem?
I think the biggest problem with the band cells idea is that we're not seeing papers all over the place about everyone finding band cells.
Yeah.
Like there is one paper on them, and their pictures are not that pretty. Like that, it's not like a widespread phenomenon.
Mhm.
Band cells don't seem to be this big thing that's everywhere.
Right.
Where were they? Entorhinal?
Entorhinal, yeah, but like the fields don't look like that. They look like grids with like with heat with the bumps of the grids attached by little like vague heat tracks.
Well,
But it's interesting because I think about in cortex, I see a lot of stuff that looks like band cells. Your orientation preferences look like band cells, and I don't see anything that looks like grid cells. So I'm thinking cortex the whole time, right? I'm thinking like, oh, what do I see in cortex? What would be the
Can I make a quick point? The band cells are not the inputs to these grid cells, right? The band cells are a pathology of grid cells that don't have balanced input.
I
That only
understand
have
that.
one input.
I
That have
understand
one input that dominates
in this model.
in this
Yeah,
model.
and
Yes.
I was questioning that. It may be true in Toronto cortex, maybe not. But I was asking the question. That's what this model says, but could it be just in theory? Could it be that actually band cells? Could I build a system that's built on band cells and grid cells are a result of that, not the other way around? It's because it's hard. It's a little hard. It's hard to imagine individual dendrite on stellate cells having this orientation preferences. It's not impossible, but it's just harder. So I was trying to get away from that.
I mean, if these are learned phenomena, you could develop very precise pattern detectors on
You
these
could, but I don't know how you would learn it. It's just harder. I'm not saying it's impossible.
I will get to the learning and self-organization part. I just wanted to point something out, saying there's no evidence for these band cells. The input to that these neurons receive on the dendrites are not band cells, right? They are theta cells. They are highly rhythmic, and they just happen to have a theta rhythmic spiking that speeds up with movement in specific direction.
Yeah.
Which is exactly what we find when we try to categorize all these different neurons in medial entorhinal cortex. Yes, there are the stellate cells. They are not bursty, right? They are not really rhythmic because they have these firing fields and they're like silent in between all the time. So they are sparsely active, and then they don't link to a specific theta phase. They're not phase locked because they're precessing. So they occur at all kinds of different theta phases. And they have strong phase precession. So that's the cells that you see on the left. The layer 3 neurons also belong in that category. There is a separate category of cells that are highly theta rhythmic, that have faster spikes, that are bursty, that might just be the kinds of inputs that we would need to form grid cells or band cells if they're missing inputs in layer 2 stellate cells. So the required inputs are there. And the observations of band cells in this interpretation would quite simply be that these are stellate cells that did not properly balance their inputs. Which of course leads us to the question, how would they balance their input?
I just want to point out again that there's this, what's going on in the entorhinal cortex, what's going on in cortex. We think this, fundamentally they're solving the same problem, but they might be doing it different ways. And so that's some of my comments are really directed towards cortex, what we see in cortex.
Yeah, maybe it's worthwhile to point some of these things out with regard to cortical grid cells, right? So if cortical grid cells work at all, like what this oscillatory interference model describes, then you would need to hunt for cells that have, for example, an intrinsic membrane oscillations. Which cells actually do that? It turns out that the cells that have that very reliably in entorhinal cortex, where there are grid cells for spatial navigation, they're all like stellate cells. So I don't know if that means that they are stellate cells in wider neocortex that might have.
Stellate cells are known to exist in layer 4 throughout the cortex, but I think they exist elsewhere too.
Right. Do they also have a small integer number of very strong superlinearly summing dendrites? Which is another property that you would need to order to do this interference.
Maybe it isn't that way.
Right, maybe it actually works different in wider neocortex.
Yeah,
right?
you could use the same basic oscillatory mechanism, but focus a variation on the theme, not a totally different method. I like this oscillation idea. When I first heard about it, I was like, oh my god, that must be right. It just seems like it solves so many problems. It doesn't mean it's sufficient on its own, but it struck me as very, very plausible.
So one interesting and important question is how finely tuned do these directional inputs need to be to work to form this hexagonal grid in an oscillatory inference model? And it turns out not at all. So what you see here in A through B is they simply divided up inputs by like 10-degree increments. And all that they said is that, okay, I want to sample from this distribution of different inputs. So I guess that's 36 total inputs. And the only rule that they enforced is that, well, pick, what was it? 3 inputs, 6 inputs. I'm not sure exactly where it is.
It's third line down on the second column. Envelopes of 3.
Right, exactly. Envelopes of 3 inputs, right? So all of these different grids have 3 inputs, sampled from this distribution. And the one that you see on the left is the one that is best. I will explain in a second what we mean by best. And the one on the right is worst.
Yeah.
One way to address that is to simply say, okay, how many spikes do we get? The one on the left has some 1,300 spikes in this simulated travel over this grid that gets spanned. And then the one on the right, the worst one, has 433 spikes. The difference between the one on the left and the one on the right
Isn't it 220?
is that the one on the left has a perfect 60 degrees between the three different chosen angles, and the one on the right has only 20 degrees between the three different angles. So in fact, the directional inputs for the one on the right differ by only 20 degrees, which is the worst behaving model in terms of delivering the least amount of spikes. So one thing that we can learn from that is that it's very simple plasticity model that just says pick the inputs that generate the most spiking. So you simply have a learning rule that maximizes postsynaptic output. Right? We'll automatically try to converge on a 60-degree separated solution, which is a very mathematically beautiful thing about this whole oscillatory inference models. It has to do with the nature of cosines.
That explains a lot.
It would explain a ton. It means that, yeah, these fields can be degraded sometime.
Isn't that also under the assumption you have only three? If you had more than three, then would that 60-degree thing come out again or not? I don't know.
Yeah, I mean, it's the same. Yeah, the model would get better and better.
No,
right?
no, the question, the 60-degree, you're arguing that the 60-degree thing we see just comes out naturally from selecting things that give you the most spikes.
Right.
But I'm asking, I don't know yet, I have no idea, but I'm asking, that's also on the assumption that you're picking three different orientations, three
Right.
different directions. What if you had 4 or 5? You said there were like 5.
I
5.
presume you would get sharper firing fields. I don't know if
Would
it
you-
increases the
would they still be
firing.
60 degrees, or would you see something different?
You would assume that would preserve that because that maximizes firing rate.
Okay,
So why would you pick a
I
fourth
guess
one?
it wasn't clear to me that maximized the fire rate if I had 4 different directions. Or 5 different directions. That wasn't clear to me.
Right.
And then I have the extra dimensions. What do I do with them? Because earlier, it would be a lot asking a cell to just always pick 3 orientations. You follow my question? I don't know.
Yeah, I understand what you mean. I can't answer it because I didn't test this here.
Yeah, that's okay.
Right.
You can keep going. I know it's late.
So the point is that they're making even directions differing by as little as 20 degrees produce grid-like patterns with strong modulations. We have argued that, gets us to the next point. So that essentially explains what kind of plasticity mechanism you would need to learn those 60-degree orientation inputs. I want to read page 805. I'm sorry?
Are you at your 1 hour boundary right now?
Probably, yes. Just want to finish this one point. The simplest model for hexagonal packed grids involves... No, that's not the one that I mean. The left. 805. Here it is. So, if dendritic interference patterns are chosen with random directions, the frequency with which all 3 are simultaneously strongly active, i.e. their product is near its maximum, will be greatest if the 3 patterns are collinear, or else when they differ by multiples of 60 degrees from each other. Thus, self-organizing learning rules, in which plasticity is triggered by maximal or near maximal levels of postsynaptic activity, are likely to converge on irregular hexagonal grids so long as collinear inputs are excluded. So in addition to this mechanism that just simply said, whenever things make me fire, connect strong, you need a mechanism that says when 2 of my inputs are collinear, right? Then I somehow have to prune one of them. So how that would work in the microcircuit and as a plasticity rule is again an interesting question.
Does this say the Hubel and Wiesel results of the orientation bands? Does it say anything about that? Wouldn't that think those would snap to certain predefined, like 3 different orientations based on that, or is that?
Well, we haven't made that connection yet, Matt.
Yeah.
I was proposing that's an area of thinking about those orientations. That's a very speculative idea that was introduced recently. So I think it's an interesting question. You don't see that. You see these things snapping, but more in terms of not three, but many dimensions. If I look at how many different orientation columns do I get?
Right.
It might be 8 or 10.
Right. But there's a continuous nature to it through the.
Well, I think it actually snaps at 8 or 10.
Oh, degrees?
No, or 8 or 10 buckets.
Buckets. Okay.
I don't think it's continuous.
Got it.
If you go from one mini column to the next mini column, it is a 10 degree, 20 degree type of thing. It's not continuous as far as I know.
All right, the last point I want to make for today, which neatly answers 3 questions that are important, is sort of shown by this slide here. So 3 important questions that we might point out. So one is that we already pointed out that phase alignment is necessary for this to work. I showed this here, right? The different oscillators that I put onto my neuron actually need to align in phase for this to work as a proper grid cell. The second one is that the information about direction and speed isn't perfect, right? There's noise in it. So there will be drift in the model, which will destroy it. So how do you solve that problem? And then the third one is that we know that grids are reliable, suggesting things like environmental anchors, so associative learning with place cells. How do we address all of these 3 questions? Well, you can address them with one mechanism, which is shown here. If it is true that a place cell can learn to associate itself with a grid cell, which is likely given the high amount of recurrent activity and lots of associative plasticity, then if you had a mechanism such that the place cell could reset the theta phase of the theta phases of the grid cell inputs, then you would solve all of the 3 problems I just outlined in one go. Because if there's a grid cell that fires at any location, it would automatically sync up the oscillators of the dendritic subunits of the grid cell. It would get rid of the drift that has accumulated, the imprecision in the odometry of the brain, so to speak. Right? If my speed and heading are not perfectly clear, I will clear up the noise that is in my system. And I also now have a mean to anchor grid cells to environmental stimuli. Right? If environmental stimuli are linked to these place fields, there's now a learnable correlation, right, between the place fields and this linear interference pattern. That gives me the grid. They show how this works in theory in this one picture. So you have a nice hexagonal grid at first. You let the simulated agent run around, and in this case they simply added a bit of noise and uncertainty into their heading and the speed, like some normal distribution from which they are drawing their things. And you will see that the grid quickly erodes. However, if you have a mechanism such that there's also a place cell in the system that fires at this spot here, and when that cell fires, it automatically resets the theta phase of the contributing grid cell, then you will see that the grid heals itself.
It snaps to the place.
Exactly.
Somehow?
It snaps to the place cell, and it makes sure that whatever erosion there was in the problem of this oscillatory inference model is all this fine-tuning in these oscillations.
Yeah.
And that's what it's commonly criticized for. So there needs to be a mechanism to overcome that. And one mechanism to do that is to quite simply say, this network heals itself by resetting the theta phase occasionally.
Syncing to place somehow.
And these theta resets have been observed. It's not entirely clear. We don't have sort of simultaneous recording of place and grid cells where we see this causally in a behaving rodent, but we have seen theta phase jumps and resets in recordings. So since this so neatly solves 3 of the big problems I laid out, right? I think this is a very fascinating mechanism.
So we've talked about this basic idea for a long time here, that place cells, and it's not a new idea to us, place cells represent your location based on sensory input, and they can now be associated with a set of grid cells that are active. And so when you know your place by sensory perception, you can lock in the appropriate grid cells at that point in time. whether it's blocking the phases or which cells are active or whatever, the basic idea of that. What struck me is odd about this, you talked about like a single place cell being locked to a single grid cell. Was that really what they're saying, or is it a population of place cells to a population of grid cells?
Yeah, that's a really good question, and in fact that leads us to the continuous attractor model. Because the truth is, of course, that all these grid cells are interconnected, and they come in modules, and some of those have the same phase, and that is the whole continuous attractor part. To get away from the problem with the oscillatory inference model, it explains all the grid cells in isolation. The different grid cells in this model have nothing to do with each other, other than that they sample from a common pool of inputs.
So the oscillatory dynamics for the grid cells, and then a CAN model perhaps for attracting to place fields.
Yeah, and for making the different grids work in unison, and for making one place cell not just being able to reset one grid cell, but instead to realign the entire grid and all of its contributing.
In that paper when they were showing that one grid cell, that one place cell, and it had a line down to a grid
right,
cell.
you meant this.
Did they mean a population then?
No, this is not a population paper yet. This is the preceding oscillatory inference paper.
No,
so this does not have lateral.
When they say, there's that RNG place cell, and it's you talked about it as if it was a single cell, where a field actually is a bunch of place cells. If you're in location, you have a bunch of place cells active.
Right.
Were they making maybe they weren't even making this argument, maybe they're just talking about it conceptually, but you talked about it as a place cell. And did you mean that? Did the paper mean that? Or did they mean there's a place and some place cells associated with that?
Well, they make the argument that multiple grid cells, right, might learn to associate themselves with all kinds of place cells.
Okay, all right. It brought up a new idea I hadn't thought about before. I've always assumed that it's a population of place cells, it's a population of grid cells. That makes sense. Everything in the brain seems to work that way. If you're going to align a single thing with a single thing, you'd have to do it like a mini-column hypothesis. You couldn't do that with a learning.
Right. No, the good thing is that
that was introducing a new idea that I hadn't thought about before, which is that place cells are this sparse code of your place, but how they get transferred to the grid cells, I still believe it's going to be through population codes. But it was the question was, maybe there's a columnar activation going on there. If that doesn't make sense to people, don't ignore it.
No, I think I get exactly what you mean. The nice thing about this place cell firing that it occurs in this global theta cycle. And so there's really like a unique opportunity where the whole system can sort of, let's say, this opportunity, right, I'm going to use to reset the phases.
What is left that's really unexplained that we're going to next time talk about the attractor problem?
Maybe it's the last thing we do, just go through this list, right? So we have a model that produces a nice grid, and it has a stable scale because there's a stable theta. You can argue that if you have a reset mechanism that will, that's going to give you stable offsets too. What's not exactly clear is how the grid distortions would work. Like there's stuff with novelty of environment, it expands and then goes back but maybe with like place cells, you can do something here.
Yeah, I think
right?
that's what the answer is.
So that's the same thing with the transient global grid upscaling. One way to do that in the model is to just change theta. So I would say there's a mechanism to do this, but it's not really clear if the brain does it that way. There is clearly now a role for the place cells and the remapping.
Yeah, okay.
Same with like environmental anchoring. The independence or dependence of grid cells and place cells is somehow addressed in this model in the sense that they don't rely on each other, but they self-stabilize each other, right? Yeah. So I will put a tentative check mark there. We address the local field potential, this dominant theta. We have a role for this theta algorithmicity. In fact, we are postulating that these theta cells are the important inputs to the dendritic subunits. Obviously, the model was built to explain this whole thing about theta precession, so the speed up of the dendritic oscillation versus the somatic main theta. It can, and in fact has an important mechanism for this theta phase resets, which are important to keep the whole system working. It turns out if you build a computational model, it's not very hard to make the firing rates for this work out. Burstiness is a different question. Intrinsic membrane potential oscillations are intrinsically assumed in all of these models, right? They have a somatic intrinsic membrane potential oscillation. The theta correlates to speed and novelty I didn't really talk about yet, but it is quite easy to show that when you move faster and you just modulate the main theta, the omega s, that you get changes to it, but yeah, we didn't really talk about it. Nor did we talk about the novelty, even though there is a tentative mechanism for it. Lots and lots of fast plasticity, right, is what you would need for this to work, for it to choose the right inputs with this learning rule, self-organizing to produce maximum firing rate, and also lots and lots of associative learning between place cells and grid cells to make this.
I'm going to go back to my question. So it's sort of obvious that almost all these things are covered. My question was, what isn't covered?
Right. So, for example,
let's start with that. Let's go. What are the big things that it's not doing?
Right. So, what is it not doing? So, for example, it does not talk at all about the most dominant pattern of connectivity for these grid cells, which is the lateral disynaptic inhibition, which is super powerful, super reliable, and in this model, there's zero interaction between the grid cells. Each grid cell is by its own.
Okay, right? So but that's not a functional loss. That is we have something going on. We don't. That's a biological mechanism that's not working. But it's not like I mean, in terms of what we're trying to achieve functionally, that's the left column. It's achieved all that, right?
Yeah, most of it.
So
yeah.
So again, why are we going to be introduced? Is there a top line, one word, one sentence answer why we have to introduce continuous attractors?
Continuous attractors. Yes. I think the number one reason is that continuous attractors give us a lot of stability. So it's like I think it's really there.
But then the stability can come all from the place cells.
Right, but setting this up in an actual model of like spiking neurons rather than a theoretical model like this, right, turns out to be really hard, and it gets very easy if you can use lateral connections, which there are plenty of.
Well, lateral connection doesn't say, you have this, Mexican hat thing going on. It doesn't. That's something else.
Mm-hmm. No, I think it's not really a Mexican hat because it's like an inverse Mexican hat, right? There's no excitatory connection. It's a weird attractor that doesn't have.
Attractor models were a reason. They were using attractor models to try to explain the grittiness of the grid cells.
Right.
But we've explained all the grittiness of the grid cells.
You don't need that.
You don't need attractor models for that. So if you're saying, the oscillatory model does great, what we're missing is some sort of stability, we're going to use disynaptic inhibition to do that. That is not merging with an attractor model, as far as I'm concerned.
I'm not sure I understand. Like it's...
The attractor model on its own explains grids.
Oh yeah, sure.
But we don't need that anymore.
Well, it doesn't explain things like the theta precession, for example, very well, right?
I know. That's why my point was when you started off this talk saying I'm going to show that we need a hybrid model for both of these things. And I don't understand. I already, a long time ago, abandoned the attractor model in favor of this one because this seemed to be the right answer. So now why are you going to, and you just showed that it's the right answer, so why are you going to introduce the attractor model? You haven't done that yet. That's going to be the next meeting, but I wanted an upfront clue, like what's missing? Why do I need to think about an attractor model? I've already, in my mind, written it off.
Yeah, I agree with you. It's a much more attractive model.
So, but why are you going to introduce it again?
Because it accounts for it can explain many of the same things. So it's the same explanation, different explanation.
But not
of the same
as well,
things.
by the way.
Well, that is people argue depending on which camp they come from.
Okay, in my opinion, not as well.
Right, exactly. So some people say they can, account for almost as many things with an attractor model. And in many ways, the attractor model is much simpler than this, right? Like this whole idea of having all these membrane oscillators at like slightly different frequencies and mechanism for phase resetting.
Okay.
Yeah?
But right now, given what we got, if we accept the oscillatory model, I do not see a need for introducing the attractor model.
So in these oscillatory models, every cell is kind of implementing a grid by itself, independent of all the other cells.
Yes.
From that, one of the most interesting things about grid cells is the fact that their spatial phase is consistent across environments.
Oh yeah.
Take two grid cells and they're consistent across environments. Do you get that with purely...
No. No,
So that's the
and
answer to
all,
your
all
question.
these global things also are not explained very well. Like the fact that when the grid changes scale, right, it changes everywhere in synchrony.
Yeah.
That works very well if you can make use of all the
Okay,
lateral connectivity.
So that's the thing. That was my question earlier. Thank you, Marcus. Was like, what's missing? And by the way, I don't know if I have to use Attractor, the Attractor model to fill in those missing things. There might be other ways of doing that.
That's a key missing part. From
Yeah, the thing that was, I was struck by Mike Hasselmo's comment to me personally when I showed him that David Tank paper and I showed him the scale at which these grid cell modules exist. And he said, holy, whatever word he used, there's no way that could be done with an Attractor model. And I said, why do you say this? There's not enough cells there. It's not enough space to do it. It's just not going to be possible.
Oh, interesting.
And it struck me as like, oh, he seemed pretty confident about that, and I can understand why he said that. So at that point, I kind of said, the model that was developed is probably wrong, and this has got, although it's more complicated, it just explains everything. And so now we have a few holes that are missing, and I think enumerating those as functional holes is the important thing, not saying we need to add the Attractor model.
Right.
It's like, how do we take this model and fix the few things that are missing in it? Is the question that I would prefer to look at it, as opposed to, now let's add the Attractor model to this. I don't think that's
Right.
the way to think
So
about
the
it.
argument that the hybrid model makes is there are certain electrophysiological characteristics that are not adequately explained by the oscillatory inference model. They actually show that, like, very specifically in the paper, they start with an oscillatory inference model, and then they have the hybrid model, and they show the difference between
Let's
those.
go to that next time. We're out of time here.
Yes, exactly.
But I think we should put our little skeptical hat on when we use the word Attractor model, that maybe it's not really what everyone's talking about in the past. Maybe it's something in between. And I mean, it just struck me that this oscillatory thing has got so much going for it. It's
Yeah, I think so
basically
too.
be right. And so in the world of two competing basic models, in my mind, the oscillatory model won. So now we have to say, well, is there anything left from the other model we could take, or is there what's missing here, or something like that?
Right.
Okay.
Yeah. Good stuff.
So this
Well,
is good.
thank
Thank you all for your attention.
Really nice review, thank you.
Yeah.
Yes.
See if I can remember it for
I
that.
like having this chart up.
Oh yeah, all the different things we want to try to account for. Yeah.
Yeah.
Maybe we can even leave this up and take a look at what the
Yeah, right, raise
what is
that
explained
stuff,
by
raise
the
that stuff and raise
by
that
the
stuff.
continuous attractor ones, right?
We also actually let you get through most of it.
Yeah, thank you for your patience. Very nice.
We have some fans online. Florian. Thanks everybody for watching. I'm going to stop the stream. Take care. Tune in next
All right,
time.
great.